All right, so those are our urinary hormones. Okay, so now we are going to go through just a few aspects briefly of different kidney function tests that can be performed in a clinical setting. And then we'll go through our last urinary structures. That should be pretty brief, okay? So for kidney function tests, and this should help you with your extra credit case study because it's going to be on the urinary system. All right, let's talk about the different ones. So first, let's talk about blood tests that can give you information about kidney function. So there's two blood tests. So you can do a blood urea nitrogen. It's called a BUN. And what this is going to measure is the urea levels in the blood. All right, so urea levels in the blood. And so they should not be elevated, right? So the normal value is around 8 to 25 milligrams per deciliter. Okay, and so if that BUN is elevated, that tells you that the kidney is not functioning properly. So the job of the kidney is to get rid of that urea, that nitrogen waste. And so if you have an accumulation of this urea, that means that the kidneys aren't functioning very efficiently. Okay, so if it's above 25, then you would want to look at what's going on with the kidneys. All right, and so then from the blood, you can also test the creatinine levels. All right, so again, like the urea should be eliminated. This is a waste product. Right, so essentially with both of these, you're just testing to see how efficiently that kidney is getting rid of waste. And so it should be less than 1 milligram per deciliter. Right, that would be a normal level. Okay, good. So that's in the blood. All right, and so then let's talk about urine. So with a urine sample, you can test quite a few different things. All right, so first you can test, and this kind of is blood and urine. All right, you can test clearance. And so what clearance measures is it uses blood and urine. All right, so it uses blood and urine. Okay, and it tells you the time to clear or remove something from the plasma. All right, so it's going to be the time to clear, right, or remove something from the blood. Okay. All right, and so usually it should take maybe like a minute to clear something, and so frequently there's a plant polysaccharide that'll be given, it'll be injected, and then you can monitor the urine and the blood to see how long it takes that level of that polysaccharide to decrease in the blood and increase in the urine. Okay, so that would be the time, that would be the clearance, so the time it takes for that polysaccharide, inulin is what it's called, to move from the blood into the urine. That is going to be clearance, okay? And so it's going to measure glomerular filtration rate. Okay, so we'll say it's a measure of glomerular filtration rate. Okay, so how long it takes something that's injected into the blood to end up in the urine. All right, so then also you can, with urine, when you get a urine sample, you can, and you should, first observe the color and the clarity, okay? And so urine usually has like a yellow color. The intensity of that yellow color would tell you something about hydration, right? How dilute or how concentrated the urine is. The actual pigment in urine comes from a molecule called urochrome. I feel like I'm losing my voice. I've recorded a bunch of videos today. Hold on. Let me get some water. Okay. A little better. All right, so urochrome is the pigment in urine, and it comes from hemoglobin. All right, so it's pigment that gives urine its yellow color, and it comes from hemoglobin, as most of our pigments seem to, right? Okay, so urochrome is the pigment, all right, and it gives it a yellow color. So urine can take on an abnormal color for many different reasons. There can be bile pigment in the urine, right? If there is bile pigment backing up in the blood, that can end up in the urine. Certain foods can change the color of urine. Certain drugs can change the color of urine. And blood, if that's present in urine, obviously would give it kind of a reddish or orange hue from blood being in the urine. And then clarity, all right, would be, is it clear versus cloudy? And so cloudy, among other things, could indicate some sort of infection. All right. It can also become cloudy if there's, more solutes and that kind of thing present in the urine. So it doesn't necessarily mean an infection, but it can indicate that. Okay. And so then odor is not something that is commonly tested by like a lab. If you send a urine sample out for urinalysis, they're not going to be testing odor. But it is interesting to note that certain foods and drugs and some conditions can change the odor of the urine. All right. So for example, the classic example is diabetes mellitus can cause urine to have kind of a fruity smell to it. Okay, so it is worth noting that urine can take on different odors based on, different things that are present in the urine. But that is not something you're going to get back in a urinalysis report from a lab. Okay, so pH, all right, should normally be a little acidic, right? So it should be around 6. And so it'll vary based on the diet and the different things that are in the urine. All right. And then you can have, you can measure the specific gravity of the urine. So this just measures the solutes in the urine. All right, so solutes in urine. It doesn't tell you what the solutes are. All right, it just tells you, are there a lot or are there not very many solutes in the urine? So, for example, pure water, all right, deionized water, just only H2O molecules, pure water, has a specific gravity of 1. Okay, so a specific gravity of 1 means there are no solutes at all present. Okay, and so for urine, normally, all right, normal would be around 1.035 or less. Okay, and when you start creeping above that 0.035, you would want to kind of look into what solutes are present there, what's elevating that specific gravity. Okay, so then you can also test urine for the presence of glucose and amino acids, right? So they should be reabsorbed, so you wouldn't expect to see them under normal conditions. All right, so they should be reabsorbed. Also, red blood cells, white blood cells, bacteria, under normal circumstances, you would not expect to see them in urine. So you can, make a microscope slide to look at those. All right, so put some urine on a slide and actually look at it and look for red blood cells and white blood cells and bacteria. You can also look for crystals, right? So formation of crystals can be seen through a microscope. Okay, and then you can test the urine for protein. And so protein should not be in urine, all right? So it should not pass that filtration membrane. All right, it should not pass filtration membrane. Okay, because generally most proteins are going to be too big to make it past that filtration membrane. Remember those filtration slits and the simple squamous of the capillaries and the glomerulus. Okay, however, if there is protein in the urine, that is called albuminuria. All right, that's just albumin, because typically that is going to be the protein that makes it through, right? If you get protein in the urine, it's going to be albumin in the urine. Okay, and so if there's damage to that filtration membrane, you'll end up seeing albumin in the urine. Okay, and then lastly, one of the other things you can detect in urine is ketones. And so ketonuria, right, is just ketones in the urine. And you may remember from our digestive chapter, right, that indicates fat metabolism. Okay, so this would be from fat metabolism. And so that could have different causes. All right, so fat metabolism could just simply be from somebody who is successfully dieting, or it could indicate something more serious like diabetes, all right, where the body is having to use fat for energy because it's not able to actually absorb glucose that's in the blood. Okay, so if you do see ketones, it's important to ask, why are the ketones there? Is it normal, or is there something else going on? All right, and so lastly, we are going to go through our final kind of structures of the urinary system. We've spent all our time in the kidneys with the nephrons. And so now once the urine is finally formed, all right, and it leaves the kidney, it leaves there through the ureter. And so the ureters, coming one from each kidney, travel to the bladder. They're each about 12 inches long, all right, and they travel through the actual muscle wall of the bladder. And so as that bladder fills with urine, what happens is it kind of like, as it expands, it kind of acts as a valve and seals off that ureter. Okay, so there's kind of like this built-in valve system. So as the bladder fills up, it shuts off the ureter, and so that prevents the bladder from overfilling. Okay, and it also prevents backflow of urine back up towards the kidney. All right, so then the mucosa, okay, so in the histology, all right, the mucosa contains transitional epithelium. All right, so when you looked at transitional epithelium in A&P1, you probably looked at a slide of the ureter. All right, that's the typical slide that's made for that tissue. So it contains transitional epithelium, which functions to stretch, right? So these ureters are made to stretch to some extent, okay? And then they do have a muscularis externa. Actually, let me put this down here. All right, so muscularis externa. Okay, and so the muscularis externa has, as we've seen in the digestive system, 2 layers of smooth muscle. It's a little different in the ureter. So it has an inner longitudinal, right? It's flipped, right? Inner longitudinal and an outer circular. So it's opposite of what we've seen before. That's in the ureter, okay? And then it is retroperitoneal, right? So it's the U in sad pucker. So it's retroperitoneal. That means it has adventitia around it. Adventitia, okay? Because it's retroperitoneal. All right, so then from the ureter, the urine would enter the bladder. Okay, the bladder can hold approximately 500 milliliters of urine. All right, so about 500 milliliters of urine. It is made to stretch, so it contains rugae, just like the stomach, and those rugae allow the bladder to expand as it fills with urine. So for the histology, just like the ureter, it's going to contain transitional epithelium. Okay. That's going to be in the mucosa. The muscularis externa is going to be highly muscular. All right, so the muscularis externa. All right, it is going to contain a layer of muscle that is called the detrusor muscle. All right, so the detrusor muscle is the special name given to the muscular wall of the bladder. It contains 3 layers of smooth muscle. All right, so 3 layers of smooth muscle. Okay. All right, and so that is just there to help contract the bladder to force out all of the urine, all right, during urination. And so then the bladder itself is going to be intraperitoneal. So it is going to contain visceral peritoneum or serosa, right? But it's only going to contain it on its superior surface, okay? So it contains, let's say, we'll write this here, right? So serosa on the superior surface. All right, and then it's going to contain adventitia around the inferior portion. All right, so the remaining part is adventitia. Okay. All right, so let's talk about for just a moment micturition. So micturition is just the process of urination, right? So micturition just means urination. Okay, and so the way micturition occurs is that the bladder is going to stretch as it fills with urine. All right, so the bladder stretches. Okay, and as occurs so often in everything we've been looking at, that stretch is going to activate stretch receptors. All right, so and those receptors are going to send a signal through the sacral spinal cord. All right, so sacral spinal cord. Okay, and that is going to activate the parasympathetic nervous system and inhibit the sympathetic. All right, so it's going to activate the parasympathetic nervous system. Okay. And then that is going to, the parasympathetic nervous system is going to stimulate the detrusor muscle. All right, so stimulates that detrusor muscle in the wall of the bladder. All right, so it's going to cause it to contract. Okay, and so when that detrusor muscle contracts, it's going to correspond to an opening of the, right, so we're going to open the internal urethral sphincter. All right. So that's going to correspond with contraction of the detrusor muscle. That internal sphincter is smooth muscle. And so then the final step of micturition is going to be the relaxation or the opening of that external urethral sphincter. All right, so then these two would be precursors for opening of the external urethral sphincter, which is skeletal muscle. All right, so voluntary control. Okay. Good, and so then that is micturition or urination. So those are the steps, right? The micturition reflex. Okay, and so then 2 terms associated with that would be incontinence. So incontinence is the inability to hold the urine. Right, so That's kind of a loss of bladder control, would be incontinence. And then retention is an inability to empty the bladder. All right, so this would be inability to empty the bladder. Okay, and so this is very frequently seen in men when their prostate enlarges, and we'll kind of look at when we look in the next chapter where the prostate is located, right? It sits around the urethra. And so as the prostate enlarges, it becomes much more difficult to empty the bladder because it's going to be pressing on that urethra, preventing it from opening fully. Okay, so that's incontinence and retention. All right, and then our very last structure is the urethra. Okay, so the urethra is going to be different in males versus females. Okay, and so in females, it is much shorter, right? So it's about 1.5 inches long. Okay, so it's much, much shorter compared to males where it can be about 8 inches long. Okay, and so that actually is one reason urinary tract infections are so much more common in women, is because of that much shorter urethra. So it's much shorter path for bacteria to get from the external opening into the bladder, causing an infection compared to males, all right, which have a much longer urethra. So it's much more difficult for bacteria to reach the urine in the bladder. Okay, and so the male urethra is divided into 3 different sections. So you have the prostatic urethra, which goes through the prostate. All right. So the prostatic urethra goes through the prostate, and then you have the membranous urethra, all right, so that it leaves the prostate and then goes to the beginning of the penis. Okay, so it leaves prostate to the penis. It's a very, very short segment of the urethra, all right? And then you have the spongy urethra, or the penile urethra, that travels through the penis. Okay, all right. So now let's just talk about histology, all right, and then we'll be done. So the histology of the urethra, the epithelium it's going to contain is going to be transitional when it's close to the bladder. All right, so it's going to be transitional near the bladder. And then it is going to change to pseudostratified. All right, so pseudo-stratified, all right, and then to stratified squamous when we're near the external opening. So it kind of morphs, it transitions as we go from the more internal portion of the urethra to the external opening. Okay. All right, and so then it does contain some, like a muscularis externa, a layer of smooth muscle. All right, and so that wraps up our urinary system chapter, okay? And so our next chapter is going to be on the reproductive system, all right? And so we will do that on our next video, all right? And I will talk to you guys then.